Hello students, today's discussion topic is mechanism of drug action. I will share you a PPT for that, and this is the PPT. Just wait for a while. Yes, I think all of you are visible now. The topic is mechanism of drug action. So what is drug action? Basically, if we talking about this introduction portion, then we will see the mechanism of drug action related with the pharmacodynamic effect. Okay. So what is pharmacodynamic? So pharmacodynamic is the study of drug effect, and it starts with describing what the drugs do and goes on to explain how they do it. Okay. Thus, easily we are talking that it attempts to elucidate the complete action effect sequence and the dose effect relationship and modification of the action of the drug by another drug is also an aspect of pharmacodynamics. Okay, so basically pharmacodynamics means study of drug effects, and this is also means the one drug actions modified by the drug actions by another drug. Okay, if we talking about the principle of drug actions, thus the drugs do not impart new functions to any system, organ, or cells. They only alter the pace of ongoing activity. Clear? However, this alone can have profound medicinal as well as toxicological impacts. But we are not talking about the gene-based drugs. Means those drugs who are gene-based, they are not categorized in this kind of principle. Okay. The basic types of drug action can broadly classified as, you can easily see, first one is stimulation, second is depression, third one is irritation, fourth one is replacement, fifth one is cytotoxic action, sixth one is antimicrobial action, and last one is the modification of immune status. If we talking about the first types of drug action, means first one is the stimulations. Then we can easily elaborate in this way that stimulations, it refers to selective enhancement of the level of activity of specified cells, like adrenaline stimulate heart, pilocarpine stimulates salivary glands. However, excessive stimulation is often followed by depression of that function. Means high dose of picotoxin, simply take an example of picotoxin. High dose of picotoxin, which is a central nervous system stimulant, produces convulsion followed by coma and respiratory depression. Clear? Then second type of classification is, that is the depressions. It means selective diminutions of actively or selective diminutions of activity of specialized cells such as barbiturate depress CNS, quinidine depress heart, omeprazole depress gastric acid secretion. And certain drugs stimulate one type of cell but depress the other, like acetylcholine stimulate intestinal smooth muscle, but depress SN node. Okay, in the heart. Thus, most drug cannot be simply classed as stimulant or depressant. So, here is another type of action, that is irritations. This cannot a non-selective, often noxious effect. It is particularly applied to less specialized cells such as epithelium, connective tissue. Strong irritation results in inflammation, corrosion, necrosis, and morphological damages. These may result in diminutions or loss of functions. Clear? Another type is, that is the replacement. What is replacement effect? This refers to the use of natural metabolites like hormones or any kind of hormones or their congeners in deficiency states, such as if we're talking about insulin, it is the replacement in it is given for the diabetes mellitus. If you're talking about the disease Parkinsonism, then we can easily given, that is the levodopa. In case of anemia, iron is another example. So these are the replacement. Okay. Then cytotoxic actions. Selective cytotoxic action on invading parasites or cancer cells, attenuating them without significantly affecting the host cell, is utilized for cure palliation of infections and neoplasms, such as penicillin, chloroquine. Okay, these are the all are the example of that. Then we are talking, if we are talking about another action, that is the antimicrobial effect, then we can easily say that it can prevent, arrest, or eradications of the infections, and infections caused by obviously any kind of microbial agents like bacteria, virus, or fungal. If we're talking about bacterial infection, then we can easily elaborate with antibiotics. Okay. And last type of drug action is the modifications of immune system, that is enhancing or depressing the immune status, such as vaccines, sera. And another drug is the levimazole, which is a immunostimulatory agent, corticosteroid, etc., etc. Apart from this, if we're talking about mechanism of drug action, only a handful of drugs act by virtue of their simple physical or chemical properties. Examples are bulk laxative such as isabgula, dimethicone, petroleum jelly, para-aminobenzoic acid, which absorb UV rays, etc., etc. And majority of the drugs produce their effect by interacting with a discrete target biomolecules, which is usually a protein. If we talk about this slides, then we can easily see that the majority of the drugs produce their effect by interacting with discrete targets, biomolecules, which is usually a protein, and such mechanism confers selectivity of the action of the drug. Functional protein that are the targets of the drug actions can be grouped into four major categories. Okay, and what are they? That is, the first one is the enzyme, second one is the ion channels, third one is the transporter, and fourth one is the receptor. Okay, means majority of the interactions with discrete target molecules is the protein. So if we categorize this protein in four major parts, that is the enzymes, ion channels, transporter, and receptor also. Then first type of interactions with proteins or protein drug interactions is the enzyme. Okay. So under the enzymatic actions, we can say almost all biological reactions are carried out under catalytic influence of enzyme. Hence, enzymes are very important target of drug actions. Drug can either increase or decrease the rate of enzymatically mediated reactions. However, in physical system of enzyme activities are often optimally set. The stimulation of enzymes by drugs that are truly foreign substance is unusual. And this kind of enzymatic actions are categorized by two types of action. First one is the enzyme stimulations and second one is the enzyme inhibitions. Okay. If we're talking about the enzyme stimulation, then we also, this is also relevant to some natural metabolites only, such as pyridoxines. Pyridoxine act as a cofactor and increases their decarboxylase activity. And several enzymes are stimulated through receptors and secondary messengers, such as adrenaline stimulate hepatic glycogen phosphorylase through beta receptors and cyclic AMP. Stimulation of an enzyme increases its affinity for the substrate so that rate constant, which is called as Km of the reactions, is lowered. Okay. Apparently, increase in enzymatic activity can also occur enzyme inductions. Okay. If we talking about this slide, that is the stimulation unusual with foreign substance occurs with endogenous ones, and I have also discussed about adrenaline and their stimulatory effect. And so what is enzyme inductions? Such as apparently increase of the enzyme activity can also occur by enzyme inductions, such as synthesis of more enzyme protein. This cannot be called stimulation because the Km does not change. Okay. And many drug induces this microsomal enzymatic activity. Okay. Then, if we talking about the enzyme inhibition activity, okay, we are talking about enzyme inhibitory activity. Okay, this is a important topics of discussion, that is the enzyme inhibitions. Okay, basically, if you see, this is categorized as two simple steps. First one is the non-specific and another one the specific. Okay, if you see some chemicals such as heavy metal, strong acid, alkalis, formaldehyde, phenol, etc., denaturate the protein and inhibit all enzymes non-selectively. Okay, this kind of enzymatic actions are the non-specific, and they have limited medicinal value, restricted to external application only. If you're talking about selective or specific, enzyme inhibitory activity, then we can see or we can elaborate, it is the common mode of drug action, whereas non-specific enzymatic activity are not. But selective or specific enzymatic inhibitions are the common mode of drug actions. Okay. Such inhibitions is either it may be competitive or it may be non-competitive. Okay. If we're talking about competitive actions, or it is also generally called, that is the equilibrium type, then you can say the drug being structurally similar, that is the competes. Okay, I will show you the slide, this slides. Okay, you can easily correlate with it. Okay, if we're talking about the competitive or equilibrium type, then the drug is being structurally similar, competes with the normal substrate for the catalytic binding sites of the enzyme. We know that enzyme has two sides. One is the catalytic binding site, which is called is the active site, and another is the allosteric site. Okay. So if the drug being structurally similar, okay, competes with the normally substrate for the catalytic binding sites of the enzyme so that product is not formed, which is shown in this figure. Now, a new equilibrium is achieved in this presence of the drug. Such inhibitor increase the Km, but Vmax remains unchanged. Such as higher concentration of the substrate is required to achieve half of the maximal reaction velocity, but if substrate concentration is sufficiently increased, it can be displaced the inhibitor and the same maximal reaction velocity can be achieved. Clear? Examples. I will give in the examples. First one is the equilibrium type. I'm repeated this, that is the equilibrium type. If the drug is structurally similar and which competes with the normal substrate for the catalytic binding site of the enzyme so that the product is not formed or non-functional product is formed and new equilibrium is achieved in this presence of the drug, such inhibitor increases the Km but Vmax remains unchanged. That's why it is called equilibrium. Why it is called equilibrium type? Why? Because if higher concentration of the substrate is required to achieve half maximal reaction velocity, but if substrate concentration is sufficiently increased, it can displace. That's why this is called as equilibrium type competitive enzyme inhibition. Clear? Then, if we're talking about these are the examples. That is the cholinesterase enzyme. Substance is the acetylcholine. Drug is the physostigmine neostigmine. The drug displays the acetylcholine. Then we see the effect of anticholinesterase activity of the physostigmine and neostigmine, and same like actions of the sulfonamides, which displays the PABA, and enzyme is the folate synthase. Okay, these are a lot of the examples. And what is non-equilibrium type? Now, non-equilibrium type enzyme inhibition can also occur with drug which react with the same catalytic site of the enzyme, but either from strong covalent bond or have such affinity for the enzyme that the normal substrate is not able to displace the inhibitor. Okay, that normal substrate not able to displace the inhibitor. Such an example, organophosphorus compound. If we talking about neostigmine and physostigmine, these all are the carbamate compounds. Okay, which are the example of equilibrium type. If we're talking about the non-equilibrium type, organophosphorus compound are also a anticholinesterase drug or else anticholinesterase agent, but which is not able to displace the inhibitor. Okay. So first one is the organophosphorate, and second one the methotrexate, which is an anti-cancer drug that has 50,000 times higher affinity for the dihydroxyapatite reductase than the normal substrate DHFA. Okay. This is the non-equilibrium type. Okay. So in the enzyme inhibition process, there is the there is the competitive type, and under the competitive type there is the two type, that is the equilibrium and another is the non-equilibrium. Now I'm coming with the non-competitive type. Just here the inhibitor reacts with an adjacent site and not with the catalytic site. Okay. I have previously told that enzyme has two sites. One is active site, and which is also referred as catalytic site, and another is allosterase site. But in non-competitive inhibition, the inhibitor reacts with their adjacent site and not with the catalytic site, but alters the enzyme in such a way that it loses the catalytic property. Thus, Km is unchanged, but Vmax is reduced in this case. An example of this kind of enzyme inhibitions, such as acetazolamide, which is a carbonic anhydrase inhibitor, and here the enzyme is the carbonic anhydrase. And another example is the aspirin indomethagen, where the enzyme is the cyclooxygenase. Okay. Now, the second type of compound is the ion channels. Protein which act on ion selective channels participate the transmembrane signaling and regulate intercellular ionic composition. This makes them a common target of drug actions. And drug can affect ion channels, some of which actually are receptors because they are operated by specific signal molecules and either directly and called ligand-gated channels. Okay. Such as if we talking about some kind of examples, that is quinidine blocks myocardial Na+ channels. Nifedipine blocks L-type voltage gated sensitive Ca2+ channels. As we see in the pictures, this is the kind of examples of the ion channels. And third type of protein is the transporter. Okay. These are the ion channels, example of the ion channels, which we discussed that the nifedipine blocks the L-type voltage gated sensitive Ca2+ channel. Okay. And third types is the transporters, as you see in the picture, that the several substrate are translocated across the membrane by binding to specific transporter or carriers, which either facilitate diffusion in the direction or concentrate gradient or pump the metabolic ion against concentration gradient using metabolic energy. Okay. Many drugs produce their actions by directly interacting the solute carrier, that is the class of transporter proteins, to inhibit the ongoing and physiological transport, as we see in the picture. Okay. If we are talking about some kind of example, then we see that is the fluoxetine inhibit neuronal reuptake of 5-HT by interacting with serotonin transporter, that is the ACRT. Okay. These are the examples of the transporter. And last mode of the drug actions and protein, which is generally the major protein, which is generally involved, that is the receptors. And what is the receptors? The largest number of drugs do not bind directly to the effectors, such as enzymes, channels, transporter, structural proteins, etc., but act through specific regulatory macromolecules which control the above listed effectors. Okay, these regulatory macromolecules are the sites on them which associated with the interact with the drug called receptors. Okay, if we define the receptor, and receptor is defined as a macromolecule or binding site located on the surface or inside the effector cells that serves to recognize the signal molecules and initiate the response to it, but itself has no other functions. Okay. If you're talking about some drug receptor interaction, then we should talking about some kind of terms, that is the agonist, antagonist, inverse agonist, okay, partial agonists, etc., etc. Then if you're talking about the agonist or antagonist, then we should discuss about the 2 points, that is, one is selectivity and another is the affinity. And what is selectivity? Selectivity is the binding of the drug to the receptor depends on their physiochemical structure. And what is affinity? Affinity is the strength of binding between the drug and receptor, means the ability to bind. This is called as affinity. And I think selectivity and affinity is present all kind of substance which are involved in the drug receptor interaction. And another term is the efficacy or intrinsic activity. What is this? Now, efficacy or intrinsic activity is the ability of the drug to elicit a pharmacological response after its interaction with the receptors. That's all. Okay. And if we talking about this terminology, that is the agonist, antagonist, partial agonist, inverse agonist, then we should say agonist means which have the affinity or selectivity and also have the intrinsic activity. Antagonist means which have the selectivity or affinity, but there is no intrinsic activity, means IA. If we intrinsic activity is summarized as IA, then IA is 0 and affinity is 1. And what for partial agonist? Now this is which have the affinity but intrinsic activity between 0 to 1. What is inverse agonist? This is also have the affinity and selectivity, but the intrinsic activity is -1. That's all. Okay, I think all of you understand the concept of the mechanism of drug actions. Okay, thank you. Bye bye.